Question: UV detector. ANSA UV detector is an inline device that measures the UV absorbance of the HPLC eluent and provides a continuous signal that can be used to quantify the amount of chromophoric compounds, ethylene, acetylene, carbonyls, acids, esters, nitrile groups, emerging from the HPLC column. There are three types of UV detectors. 1. Fixed wavelength. 2. Variable wavelength detectors, VWD, or UV vis absorbance detector. 3. Photodiode array detectors, PDA, or diode array detector, DAD. 1. Fixed wavelength UV detectors with low-pressure mercury lamps were first in the late 1960s. Fixed wavelength UV detectors are found mostly in low-cost or portable systems. 2. Variable wavelength detector, VWD. This device uses a deuterium source and a monochromator to allow the selection of a particular wavelength in UV vis region for selective detection. 3. Photodiode array detector, PDA. This is a common UV detector that monitors the entire UV vis spectrum of material passing through the flow cell using a photodiode imaging sensor, typically consisting of 512 or 1024 pixels or elements. The detector yields both absorbance and spectral data that can be used for quantitation, identification, and peak purity assessments. Limitations. 1. Analytes must have chromophoric activity. 2. The mobile phase must be transparent to UV to have acceptable sensitivity and linearity. 3. Detector responses depend on E and can be highly variable for different compounds. Advantage: non-destructive, very reliable, and easy to use. Medium to high sensitivity to compounds with high molar absorptivity. Detection of organics at low UV wavelength, compatible with gradient analysis. UV vis detection is valuable tool in pharmaceutical industry, biopharmaceutical, biotech, PET isotope research and production, food and beverage, oil and gas, adhesive, lubricants, detergents, cannabis, hemp, marijuana. Quality control labs, quality assurance lab.